Okay. All right. So good morning, and it's not yet 8. I think we just maybe we'll give a minute or two, and then we will start. So in the meantime, let me remind you all that you have a mini project to submit. I think the deadline is 7th by 9 a.m. So please be on time. There will be no further extension of this deadline. And if you have any issues in doing the project, if you do not understand how you should be doing this or anywhere you're stuck, please get in touch with me. If you've not begun doing the project, let me warn you that it is not something that you can finish in an hour or 3 hours. It would take a considerable amount of time, so please start doing the project if you've not already begun. Okay, let me see what's the time. It's 8. Okay, so I have shared a screen. Is that visible to all?
Yes, ma'am, it is
Yes,
visible.
ma'am.
All right. And have you all received your marked answer scripts?
Yes, ma'am.
Yes, ma'am.
Okay. Do you any of you have any issues with the marking scheme? Anything that you would like to, if there is anything, because I've sent individual sheets to everybody. So if you have any issues, anything with the marking scheme or anything to contest in the marks that I've allotted, please get back to me. Okay, so we'll start. Um, so in the last class, we have been looking at bacterial motion and bacterial flagella, which powers and propels bacteria through a liquid medium. And excuse me. And what we learned the last time was that so the bacterial flagella system is very much like the motors that Tesla designed and became famous for, in the sense that they have something called a motor and a stator part, and so the analogy is very striking. But if you compare now the motor that runs our car engines with the motor that propels the bacteria, you will see that the bacterial flagella has a very high rotation per minute or RPM compared to a car. And this is and the speed at which the bacteria can propel itself is, if you give an analogy, it's like as if I'm running 20 times my body length per second. Now that's tremendous, right? 20 times my body length per second. So that's nearly the speed at which these bacteria get propelled by flagella. And what makes it even more remarkable is that while I'm thinking of 20 times my body length per second in air, and even the fastest moving cars that we have move in the medium that has air in it, not water. These bacteria are actually moving through a very viscous medium, and it's not just water, it could be even, say, blood, right? If when there is sepsis and bacteria actually invade the circulatory system, they're moving through blood, or it could be any other viscous medium. For example, if you think of the bacteria that you're growing in laboratories, you're growing them in liquid medium, right? You grow them in something called a luria broth or in some sort of broth, a tryptone broth or some broth. Now that broth, if you look at its viscosity, that would be much more viscous than even water. But the bacteria are able to propel themselves in these viscous mediums. So that makes the achievement of the flagellar motor in terms of propelling the bacteria even more remarkable. Here we saw this picture before, so it's an electron microscopic image of bacteria with all the flagella that have been stained. And as I mentioned the last time, You could see the flagella using fluorescence microscopy, but the best way to visualize flagella and its parts is definitely electron microscope, because only the electrons when used, the wave nature of electrons when it is used to visualize these objects, it gives you the resolving power to clearly visualize what are the components inside a flagella. It cannot be done by visual field of light. You need to go towards high energy waves, and you need to go towards shorter wavelengths, higher frequency to be able to do that. Okay, and we also saw that bacteria can have different structures, and flagellar positions on bacteria can also vary. So you could have the bacteria having a one single flagella, or they could have multiple flagella. And what I missed the last time was that, so flagella is
Ma'am,
a, yes,
in the last slide, just previous slide, can you go back? Ma'am, what is the difference between like family 3 and family 2 here?
family 2 would have a polar flagella, so one single flagella from the end. It could be one, it could be two, it need not be just one, it could be multiple flagella but arising from one end. So see, let's simplify this. If you have, for simplicity, let's not think of a coccus, but of a bacillus, like a rod shaped. So what are the positions at which you could place a flagella, thinking that you are now God and you would like to place the flagella? You could place it throughout the body. You could place it at the ends, at one end. You could place it only at the lateral surfaces, so this surface and this surface, right? So all of these possible combinations are actually seen in nature. So you could have, okay, let me take my phone for example. You could have flagella arising from these two ends. You could have flagella arising from the lateral surface. You could have flagella arising all throughout the surface, which is what mostly I think E. coli is. You could have it only from one end, which is what mostly Vibrio cholerae is. So positioning of the flagella can be very different. Is that okay?
Yes, ma'am, clear, clear.
Okay. All right. So with this, we come to the slide where I have already spoken about the high speed of rotation of the flagella. So flagella is a rotary motor. So what it does, it rotates. So just like Tesla's motor, which has a rotary drum, which rotates, here you too have a rotor. So the flagella has a rotary motion, right? And whip-like, we call it a whip-like rotary motion. So then there is, so if something moves laterally, we say that it has a speed of translation of so many meters, micrometers, whatever the unit of measurement of the distance is per second. Here it is rotation. So we say so many rotations per minute, or in flagella, because rotations per minute would be very huge, we can say rotations per second. So for a flagella, the rotations per second turn out to be of the orders of 100. So maybe 100 or 200 or 300 rotations per second. So something of that order. And it depends from organism to organism. Some organisms can have, some bacteria can have very high speed rotary flagella, but in general, the speed of rotation of this flagella is very high. And the flagella itself is not a single, the main unit of flagella that you see outside is, of course, a single protein, but the flagella, the entire motor, is not a single protein. It is a multi-protein, multi-subunit complex, and can have as many as up to 60 proteins that make up the entire flagella. And what I had missed the last time was that when this flagella rotates, the flagella could rotate clockwise, that is, it could go in a clockwise direction, it could go in a counterclockwise direction. Now, depending on the rotation, whether it is clockwise or counterclockwise, the bacteria can bundle all its flagella at one end. So it's easy for us to imagine now that there is a bacteria with a single flagella, and therefore, if the bacteria has to move towards an attractant or away from an attractant, then we can understand that okay, this it must be propelling itself with the flagella. So just imagine a propeller. You've all seen a propeller, right? Or you've at least seen videos of a boat or a ship with a propeller at its end, or a submarine with a propeller at its end. Have you all seen at least an image or a video? If nothing, at least the movie Titanic, where you see
Yes,
a
ma'am.
propeller, right? So you think of that, you think of a similar analogy. It's all analogous, whatever we have designed, we have actually taken from nature. So if you think of that, you have a propeller here, and depending on this propeller, so what it does is this propeller, as it rotates, it propels this organism forward, and the medium is pushed back, right? That's how it is. It's moving forward. But now you could also steer this away. But if you have such propellers all around the ship, if this is a ship and you have propellers all around, then it's counterintuitive how it would move, right? Because all the propellers would be negating themselves. ये यहाँ पे push करेगा, ये यहाँ पे push करेगा, तो ये ultimately move ही नहीं करना चाहिए. It should be static. If you think of an analogy, if you have propellers all around, then how is this going to move? If you just have it at one end, yes, it is propelled. If you have it all around, her angle say equal forces are there, then how is this going to move? It's not going to move. It's going to be placed static. So then a bacteria that has flagella all around it would end up with a similar problem, that all the flagella are moving, and say, let's suppose that all the flagella are having equal speed of rotation, such that they are all producing equal forces on this bacteria, and the net result will be the bacteria is not moving. But this does not occur. What occurs is that these flagella, then something happens called bundling. So all the flagella, if you look now at this image down here, you see all the flagella tend to end up in a, tend to bundle up together at one pole of the bacteria in such a way that now the motion produced by the flagella gives a net force in one direction. So it's a very directed motion of the bacteria. Now, this is when the bacteria is trying to move in one direction. But what if it tries to run away from something? Then the easiest thing for it to do is to not move in a direction, but rather to just jump out of the situation. It's almost like if you've seen videos of I'm sure you must have seen this. Everybody shares this on all kinds of social media. So have you seen a video of where a cat is stunned by a cucumber, thinking that the cucumber is actually a snake? Have any of you seen that video? So if you were to place a cat, and then if you stun a cat, what does the cat do? Does the cat make a turn and then run away? It does not. What it does is it literally jumps out. Or even if we see a snake, our first reaction is not to turn and run away, but to jump out. So bacteria does something similar. So when it is attracted to something, it's moving at full force. It then bundles all its flagella at one end and propels itself. But when it has to run away, what it tries to do is it tries to disengage all the flagella and do something called a tumbling motion. So you would often see the motion of bacteria being described as run and tumble. So what is this tumble? It is very analogous to a situation where we would sort of jump out of we would just jump out if we see something. If we are startled, we are just like, ah, and then we run away. But the first instinct is to jump away. So that something similar, analogous, is done by bacteria. And to do this, the flagella do not bundle up at one pole. They sort of disengage. And what what exactly happens is that the flagellar direction of rotation changes. So the direction of rotation of the flagella when it is all bundled at one end is seen to be counterclockwise. But if it is moving clockwise, then the bacterial flagella do not engage at one pole. Rather, they are all spread out throughout the body. And then this causes a three-dimensional motion of the bacteria. Do not think of it as one two-dimensional motion because it's in a solution. It's a three-dimensional motion. Now, instead of moving like this, it sort of tumbles away. So if this was propelled, now it tumbles away out of it, out of a situation. All right, let's get back. So what we were doing last time was we were having a look at the components of the bacterial flagella, and to understand the components, we were looking at a very simplistic yet realistic model of the flagella. Before we come to the model once more, I would like to emphasize this slide that I have here. So what the slide shows you is again an electron microscopic image of a flagella. And what this particular image shows you is how the flagella is embedded inside the body of a bacteria. So the flagella, the major part of the flagella is outside that you can observe under an electron microscope. So this is what you see outside. But this entire long stretch of flagella has to be embedded inside the bacteria, and this part is what helps the flagella embed into bacteria, as well as it is the motor component that actually rotates the flagella. And this is again another electron micrograph of the same thing. You're actually looking at an enlargement of this little portion here. So what are these components? And for that, let us go back to the video. Okay. So I'm going to shut down this. So what you see here is a bacteria that is being propelled by a flagella. Now this flagella could be a single flagella, could be two flagella, four flagella, but whatever they are, they would be all bundled together at one pole so that the bacteria is being propelled in the media. And the media is highly viscous. I keep repeating it, it's a highly viscous media. So it's a very different situation compared to the case where we are moving in air or a car is moving in air. Now this shows you how a bacterial flagella actually resides inside the bacterium. Now if you go inside the bacterium, what you see is this superb machinery that rotates the entire flagella. And outside what you see is called a filament. Then there is something called a hook that attaches this entire flagella to this machinery. And this looks very similar to what the motor designed by Tesla learned. So there was something called a rotor which rotates, and then there would be some things called the stator. But the stator is actually producing the energy for the motor to move. And the energy here is derived from the differences in the proton or positively charged ion gradients between the outside the cell and the inside of the cell. So inside the cell, you have less protons, less positively charged ions compared to outside. And this concentration gradient is utilized by the motor to drive the protons, to drive the positively charged ions inside the cell, to not drive, I shouldn't say drive. Drive is again an active thing where ATP is utilized. Rather, there is no ATP utilization here. What it does is it simply allows these positively charged ions to move through. And as it allows the positively charged ions to move through, it causes a rotation of this motor. So these are the stator components attached to the flagellar motor. So here you have the motor component, here you have the stator component, Here is the channel that allows the positively charged ions, it could be protons, it could be sodium ions, to move through. And because of this movement, you see the movement of the motor. Now, how this process takes place, for this we are going to zero in on two proteins called MOT A and MOT B. Now, names are not so important. What is important is what is actually happening here. So this is again a simplistic representation of the whole thing. So here you have those channel forming proteins that form the channels as well as interact with the motor. So here is the motor. Here is one part of this protein, of the stator protein component, that interacts with the motor, and here is another part that actually forms the channel. So if you now have a cartoonic representation of it, this is what it looks like. This entire thing is the stator, and this green part is the motor. This green part here is the motor. The rest of it is the stator, because that part is not rotating, but does not mean that there is no conformational change in it. By the word stator, when I say stator, do not think that there is no conformational change there. Rather, the conformational change in the stator is going to drive the rotation of this motor component. And we will see how. So you have this MOT B, MOT A. Again, this part, this light cream or yellow part is actually interacting with the motor, whereas this part forms the channel through which the ions are passing. And this part are interacting together, and this are interacting together, as you can see in the video. Very clear. Now, there are two important amino acid residues which are here, placed here, the aspartic acid. Aspartic acid having a negative charge, it's easy for you to understand how it would interact with positively charged ions, right? So this aspartic acid residues, you have two of these proteins, so two aspartic acid residues are important in transducing the positively charged ions from outside the bacteria to inside the bacteria. And as they do this, you see, as the positively charged protons or positively charged ions, it could be sodium ions, come and interact with the aspartic acid, there is a conformational change in the protein that produces a change in localization of or conformation of these associated proteins, and hence it causes a small change in angle of the motor. And as the protons are pushed into the bacterium, again there is another small conformational change. Again there is a shift in the motor. So it's a very simplistic way to do it. Nature has designed it very simply. You have a transport of sodium or protons from outside the cell to inside the cell, and every time this occurs, every time there is a translocation, every time these protons bind to the aspartic acid residue, there is a small change in conformation that causes a small change in angle of the motor. Again, when it is released inside the cell, there is another small change in conformation that pushes it back to its original conformation. Again, there is a small change in the angle of the motor. So the motor keeps rotating every time. And this causes what's like the power stroke. So the first power stroke was when the protons had bound to the aspartic acid residue. The second power stroke is again when the protons are released from the aspartic acid residue. Okay. So far, any questions? Sorry. Is it okay? Clear to everybody?
Yes, ma'am.
Yes, ma'am, it is clear.
Thank you. So next what we will see is how is this entire machinery embedded inside the bacterium. And the last time I had explained that bacteria are generally classified into two kinds, and that is classically done with the help of something called Gram staining. And why is Gram staining going to differentiate between the bacteria? Is because some bacteria have, so basically all living organisms have a lipid bilayer that covers them, right? A lipid bilayer that packages all the contents of the cell inside. But some organisms have a cell wall that is outside this lipid layer. So those organisms are plants. Plants have a cellulose layer, could be yeast, and also bacteria have this cell wall. So bacterial cell wall is made up of something called peptidoglycan. And this is what is depicted here. You have the membrane and you have a peptidoglycan. Turns out some bacteria have a very thick peptidoglycan layer, whereas some other bacteria have a thinner peptidoglycan layer, but have an additional membrane protein layer that's outside this peptidoglycan layer, additional layer. And that makes them gram-negative. So you have gram-positive with a thick peptidoglycan layer, a gram-negative with a thinner peptidoglycan layer, but an additional outer membranous layer. So the flagella for gram-positive and for gram-negative bacteria, they have to be embedded inside the membrane, the peptidoglycan, and they have to come out either through the peptidoglycan or through the outer membrane to the outside the cell. And they also need to have a machinery that is exposed to the cytoplasm. So if you look now carefully at this video, you see those components. You see that there is this 3 layers. This is being shown as a gram-negative. You have the peptidoglycan, thin peptidoglycan, inner membrane, cell membrane, outer membrane, and you have this entire machinery, main force-generating machinery that is embedded through the peptidoglycan and through the inner membrane, and you have the motor that is exposed in the cytoplasm. So this is how the entire structure would look. But then the flagella has to, as the flagella evolves or is produced, it has to penetrate all of these layers and then come outside the bacteria. And we are going to see how that is assembled. So what you're going to see here is how the flagellar assembly takes place. So the first event that takes place here is the assembly of these ring-like components. Excuse me. So the first ring-like component assembles on the bacterial membrane. The names are not that important. What is important for you here is to just understand the concepts. So you have the first layer that forms, and then you have another layer, and this layer is very important because this layer is what would interact with your stator components. Let's go back. Let's see it once more. So you have the first layer that forms in the cell membrane. So how is the flagella synthesized in the cell, in a bacterial cell? The first layer of these protein ring complex is formed. You have a second layer that forms, and this layer actually interacts with the stator components. You have a third layer that forms this bulky motor. The entire thing forms a bulky motor. And once you have assembled, or the bacteria has assembled all of this, it now assembles a protein translocase complex that allows the translocation of the flagellar components to outside the cell. Now this flagellar components are being assembled one by one from the center of this motor. So you see let's play it once more. So now this flagellar motor has been assembled. You have the stators that have been assembled. Now the flagella has to push itself through these layers of the bacterial cell wall, outer membrane, to outside the cell. And it has to form in this process the hook that holds the flagella. So initially it starts forming with the help of proteins called flagellins, which are exported outside. And you see, the last time we heard something called a helical flagella. And now you will be able to appreciate why it is helical, because the subunits of the flagella that's called the flagellin protein are actually assembled in a helical fashion. So as you see this forming, you will understand it much better. You have the subunits that keep assembling in a helical fashion, and then there is the formation of the hook, and after the hook you have the formation of the flagella. So now the flagellar proteins will be transported, and you will appreciate that for this process to take place, the flagella needs to be a hollow tube. It cannot be it cannot be a completely packed, dense cylindrical structure. It needs to be a hollow tube for the flagella to grow in this manner. And indeed, the flagella is a hollow tube. And in fact, many of the bacteriophages, they use the flagella as their entry point too. So you see this, the assembly of flagellant proteins in a helical fashion, and you see the formation of the flagella, and at the tip of the flagella is a kind of a cap, and the flagella grows, but the cap remains at the tip. So this is in a nutshell how the flagella is formed. But I hate this. Let me come back. Do I get Okay, I'll keep it here, and I hope you can see my PowerPoint screen once more.
Yes, ma'am. Yes, yes.
Okay, okay. So far what we have seen is that the flagella is powered by proton gradients, and the proton transduction is done by something called the MOT-AB complex, that is the stator. And you see here the MOT-AB complex is shown here. And then you have something called the C ring. It's not very, I mean, it's good to remember things, but even if you do not remember what is the L ring, P ring, C ring, it's fine as long as you understand the concepts. So you have a motor component here that is attached to the stator, and the every time a proton or a sodium ion attaches to the aspartate residue in the MOT-AB complex, there is a conformational change, a power stroke that makes this ring move a certain angle, or rotate for a certain angle. And that rotation translates into the rotation of the entire flagella for a certain angle, right? So this powers the rotation of the flagella. And when the protons or the sodium ions are released into the cell, that creates another power stroke. So the conformational change is reverted back, and that causes another rotation of the motor. Okay, so now the question is that if everything is driven by sodium or proton gradients, what if you do not have a gradient anymore? What if you make the outside and the inside have the same amount of protons, such that there is no point in transducing any more protons? Or what if you make the proton concentration inside a bacteria very high? What would happen to the motor? Can any of you speculate?
Ma'am, it should not move.
Yes, it should not move. So, I mean, if you have a condition where you have too many protons inside, then pushing another proton inside is kind of unfavorable. That would not happen. So, if another proton is not moving inside, or if the movement or if the diffusion is very slow, then it should be that if this is indeed powering the motor, and if you are blocking somehow the movement of protons or sodium ions from outside the cell to inside, then you would not have those conformational changes, and therefore you should block the movement of the motor. And that is what was found in a number of experiments, and I have shared with you here one such experiment. So let's have a look at this. So what they did was they took,
Ma'am,
yes,
I had a question like this L ring and P ring, etc. There were. So we will consider that also as part of motor or not?
No, 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 no. The motor is the part that is actually attached to the stator component. These components that you saw at the top are the ones that are sort of holding it in place through, as I said, there are multiple layers through which the flagella has to penetrate out. And it has to be kept in position. So it's like you have a motor, you have a stator, and you have a whip-like thing that has to come out. But the whip cannot go like this, right? Inside or through the cell membrane, not the cell membrane, through the peptidoglycan it cannot. Through the outer membrane it cannot. It has to be held in place. So those things are there to keep them in place as it moves through these layers of bacterial cell wall or the bacterial outer membrane.
Okay, ma'am. Okay. I was just like thinking like that can be considered as a starter. Uh,
So a stator is, the name stator does not just mean that it is going to be static. The name stator also means that that has to be the point where it is producing the force for motion. So for that, I'll just go back quickly, one slide. This is Tesla's motor, right? You have this rotating drum, which is the motor. You have these induction coils, which are stator. So this is static. It is static, so it is a stator, but it's not just static, it is something that is driving this motor to move. In that sense, you will call a stator should also have something to do with generating the force for for the movement of the motor. Therefore, the mot AB complex is what is found to be driving. And we will now look at how it is going to be driving. So the point is that if indeed the sodium ions or the protons, whatever the species, so here's another thing, depending on the species of bacteria, the flagella could be powered by protons, the flagella could be powered by translocation of sodium ions. So it depends on the kind of bacteria, what it chooses to bind to. Now, here they have observed a flagella that is powered by differences in gradients of these positively charged ions. And what they try to do is to raise the intracellular pH. So if you to lower the intracellular pH such that you have an excess amount of protons now present inside the bacteria. If you have excess amounts of protons, if you have excess amount of protons inside the bacteria now, then the movement of protons from outside the bacteria to inside would be hindered, right? You just have too many protons inside. So so it is really unfavorable for the system to bring more protons inside. It's like bringing it against the diffusion gradient. This would require expenditure of energy, which the flagellar system is not designed to do. It is designed to work in the direction of diffusion motion, not against the direction of diffusion motion. There is no ATP hydrolysis, no GTP hydrolysis associated with with flagella. So that is the case. If you have too many protons inside the bacteria, then you should theoretically be able to stop the transport of protons through this mot AB complex. And if you're able to stop the transport through mot AB complex, you should be able to stop the rotation of the flagella. So they tried a similar experiment, and what they did was very unique. And let's look at some small components of this design. So they took a bacteria with a flagella, and they sort of sheared the flagella so that you take away the tip of that flagellar complex. What you end up with is a flagella which has a sticky end. So if you take away the cap, the flagellar end becomes sticky. So you can attach You can simply put beads into the solution and you will see the bead has gone and attached to the tip of the flagella. Why is this important? It is important because otherwise, how are you going to visualize the motion of a bacterial flagella? You cannot literally look at motion under electron microscopy. Now, this is something that one of you has have also written in your answer script. I'd like to point out once more. You can see or observe motion using light microscopy, fluorescence microscopy, everything in the visual range. But the moment you expose something to X-rays or electrons, you cannot do that in a live sample. If you're trying that in a live sample, you'll actually be killing that sample because those are very high energy rays, right? So that if a flagella cannot be very easily visualized under a light microscope, then how do you look at the motion of a flagella? The easiest thing to do is attach beads, just like they were doing it with myosins, with kinesins, with actin. So you attach beads, and then those beads are big enough. So you follow the beads, and you know that because they are attached to the tip of the flagella. So you're literally looking at the tip of the flagella as the bead keep moving. So if the bead is moving in a circular motion, you know that it's the tip of the flagella that's describing this circular motion. So that's the first thing, first experimental design. So you take the bacteria, you shear off the flagella end so that you create a sticky end. Now if you add bead, the bead spontaneously attaches with very high affinity to these sticky ends of flagella. And now you can follow this bead as it describes a motion. While doing this, they also reduced the pH inside the bacterium by adding this sodium benzoate. Essentially, you add any organic acid, and if you add that, then once they are taken inside the cell, the pH inside the cell reduces. Now let us look at these graphs here and what they describe. So this graph here describes the pH within a bacteria. In general, you have not added anything. You just take a bacteria, you observe what is the pH inside the bacteria, and you see that it's somewhere around 7, 7.5. And at this pH, if you now look at the rotation of the flagella, you see that the rotation is kind of constant given that you are changing the external pH from say 7.5 to say about 5.5. You see that the rotation is nearly kind of constant. But if you are able to decrease the pH inside the bacteria from the 7.5, which was somewhere here, to about say 5.5, you've drastically reduced the pH. So as you reduce the pH, you're actually increasing the proton concentrations or the proton concentration inside the bacteria. As you do this, theoretically, you should be blocking diffusion of protons from outside the cell to inside because you have too many protons inside. And then theoretically, you should be blocking the rotation of flagella. And indeed, they see this. This is the speeds at which the flagellar rotations were observed. And as they kept decreasing the pH, the flagellar rotation rates came down. So this is rotation rate in hertz, and you see the rotation has come down, which goes to point out that indeed this is primarily driven by proton gradients or positively charged ion gradients. And here is another thing that they did. So if you have reduced the pH and you see that the speed of rotation of the flagella has come down, what if you now increase the pH? If you can now add some alkaline solutions and you increase the pH now inside the bacteria, what happens? And they found that if you now increase the pH, the rotation rates again recover. So this is a reversible process. This is a reversible process, which shows that why was this important to do? Why do you think it was important to show that this is reversible? Any idea? Any idea? So you have added, so you know that if you're going to increase the proton gradients, you expect that the flagellar motion would be affected because the proton gradients are primarily driving the motion of the flagella. Why is it important to see that if you now increase the pH, that you can recover the motion of or the speed of movement of the flagella? Why is it important?
To
I
know
mean,
like
because
if it is
Yeah.
increasing the pH will lead to decrease in hydrogen ion concentration inside the cell, and it will reverse the condition towards the possibility of hydrogen ion coming from outside to inside the cell in the
Yeah, that
moment of
is that
flagella.
is expected from our first principle, right? That is what we expect. But why is it important to show? I'll give you the reason. It is important to show because you want to be sure that you're just affecting the transport. Listen to this carefully. You're just affecting the transport of ions and not necessarily collapsing the entire system because, see, biological systems, proteins work at a certain pH. What is the guarantee that if I bring down the pH to 5.5, I still have those proteins intact? What if the protein complex has dissociated? It's possible. The only way to do this and to show, so it's kind of a control system. If so, when I'm saying that, see, I have changed the proton levels here. I have increased the number of protons, or I've increased the number of sodium ions, whatever the case is. And see, now the proton translocation has stopped, and so now the flagella is not rotating. If I'm a bad person, I will say, no, I don't think so. I think that's not rotating because the entire complex has dissociated, or the flagellar complex is not in the same way. The motors and stators, they're not in there. They're not connected. The pH has all changed. It's a possibility. It's a possibility. So if there is such a possibility of dissociation, then just merely increasing the pH back wouldn't help it to recover because this entire process of formation of this huge flagellar complex is a non-trivial task. So it's kind of a control experiment to show that indeed it's only a function of the transport of these protons that is affecting the rate of rotation of these flagella. It's kind of a control experiment. So when you show that it is recoverable, you show that it is only a function of movement of these protons that was tampered with in this experiment. The rest of the flagellar structure, rest of it was still fine and intact. And if you bring up the pH, if you reduce the proton concentrations now inside the cell, you can recover the rate of movement of the flagella. Now, here is another something very interesting and analogous to how they were observing movement of kinesins, dyneins, you name it, all those motors. So when they were looking at when we were studying kinesins, dyneins, or even myosins, we were talking of something called the step size, right? How many nanometers is the motor moving for every step it is taking? Now, for a rotary flagella, a step size wouldn't be the distance traversed, but would be rather the angle it subtends for every single step it takes. Because it's a rotary motor, so it should be the angle, right? Here we are not looking at the distances traversed, yet we are looking at a torque that it generates. So for a flagella or for any rotary motor, it's all about looking at the angle it moves for every movement of the stator. So what's the angle of rotation of the flagella? How much does it move? So to understand this, uh, these people, they use something very similar to what I've described previously. You shear off the end of a flagella, you make it sticky. Now you add a bead to the end of the flagella. You simply add the bead to the solution, you'll see that those will get stuck to the end of the flagella. And now you can use a bead that is fluorescent. So if you use a bead that is fluorescent and you keep imaging the fluorescence, you will see something like this. So these are individual snapshots of the fluorescent bead taken over time. And what you see is this is, so yellow and red here describes the position of the centroid of this fluorescent image. Because as I've said, you get a very blurred image of fluorescence. Uh, and it's called something called the spread function, but whatever that is. You can think of it as a Gaussian distribution of fluorescence intensity, and you take the centroid of this, and that should give you literally the position of the bead. So if you now every time look at the centroid position, then you're looking at how over time this bead has moved. And the yellow dots indicate the previous position and the red dot here indicates its current position. So if you start from this frame and you keep moving, you see the red dot is moving every time, and then it describes in the end a circular motion. So with time you can exactly see where it is and how it describes our circular motion. So in this very system, if you now plot the angle of motion of this bead, the angles it moves, say from t is equal to 0, so t is equal to t1, t2, t3, t4 and so on, with time and the angle of its rotation. So 360 degree is one full turn, 720 is two turns, and it's describing a circular motion. You get a graph like this, analogous to what we were doing for kinesins, myosins. There you had the distance traversed. Here you have an angle that is moved, right? If you now, so this looks very literally like the step sizes of kinesins or step sizes of myosins, right? There too we had seen something like this. Here, if you look at it more closely, if you zoom in on any one of this, you would see something like this. You would see structures that come up like this. And then those have to be fitted. Those have to be fitted with to obtain the step size. And what they see is that the step size is about 14 degrees, which translates to 26 steps of the flagellar motor to complete one single rotation. And this is done in terms of milliseconds. So again, it shows that flagellar rotations are very, very, very fast. So if you have 100 rotations per second, and for every rotation you have 26 steps or about 14 degrees of motion, 14-degree angle that is moved by the motor, then you can imagine how fast this motor is actually moving. Okay, so with that, I will stop this lecture. For this part, I would suggest please have a look at this paper. It's a very simple paper, but if you read it, at least you will be able to understand the concept that there is a change. First of all, the entire movement of flagella depends on two things. One is that it is translocating ions, protons, along the diffusion gradient, not away from it, not against it. So there is no ATP hydrolysis, there's no extra energy that is being provided for the movement of the flagella. It all depends on this proton gradient. If you disturb the proton gradient, the flagella wouldn't move. The second thing is that this translocation system, the stator, is linked to the motor component. And every time it translocates the ions, there is a conformational change that causes a small shift in, or causes a small change in the position of the motor. So it moves the motor, and the movement is found to be around 14 degrees or about 26 steps for an entire rotation of the flagella. Alright, so with that, I will stop today's class. If you have any questions, I'll be here for a couple of more minutes to take your questions.
Yes, ma'am. So ma'am, my question is that what assures that the pH inside the cell, like when hydrogen ion gradient reaches the threshold limit inside the cell, that no more hydrogen cannot be coming from outside to inside, then what assures that these hydrogen ion must be get out from the cell so that the integrity can be maintained?
What ensures that the hydrogen ions are pumped out of the cell?
Yes, ma'am. Yes, ma'am.
So the integrity of what is maintained?
The integrity of motor protein so that they can take hydrogen from external to internal so that the motion
This
can be
is
done.
when we are saying that the inside of the cell has attained a pH of 5.5. If you're now actively pumping whatever protons have come in outside the cell, then how is it going to retain pH 5.5?
No, ma'am. Then hydrogen ions will come from outside to inside. Then pH will drop now if inside the cell. So
Huh?
to maintain that pH of 5.5, like say for example, when the hydrogen ion will come, then pH will drop from 5.5 to lower. So to maintain at 5.5, the hydrogen ion that are excess in the cell that will
You're not
exceed the cell.
adding HCl. You're not
No,
adding
ma'am.
HCl. You're not adding a strong acid. You're adding a weak acid.
No, ma'am.
So a
I am
weak
not saying
acid.
I am not saying HCl. I am saying when it will exceed the cell, exceed the concentration of hydrogen ion in cell. When hydrogen ion will come from outside to inside, then the hydrogen ion concentration will exceed in cell, and the pH will get lower. So that hydrogen ion must be get out by the time so that the 5.5 pH can be maintained. Are you understanding what I'm saying, ma'am? Uh,
Farman, can you
yes,
explain
ma'am.
to me what is a weak acid?
Uh, Ma'am, weak acid is which have equilibrium between its hydrogen associated form and hydrogen dissociated form.
Um, and so why and how, what would be the, so what would be the pH of, say, okay, your answer lies in this. What would be the pH of zero point 0.1 molar HCl? What would be the pH of zero point 0.1 molar acetic acid? The reason that the pH inside the cell is 5.5 and doesn't go below 5.5 is because you're adding weak acids. You're not adding very strong acids that can bring down, because you, at the end of the day, you want the cell to survive, for the entire
Yes, ma'am.
flagellar machinery to survive, so that you're able to just observe the changes in the proton translocation machinery and how, or in the translocation of protons and how that translates into movement of flagella. What you're asking is that when the pH has come down to 5.5, what prohibits it from going even down further? What
Yes, ma'am.
prohibits it from going down even further is the kind of acid that you've used.
Okay.
It is the kind of acid that you've used. You've not used HCl. You've not used HNO3. You've used benzoate. You've used something that is a very weak acid.
Yes, ma'am. Yes,
That's
ma'am.
what is preventing it from going down even further. So the choice of acid, what your question is, your question again highlights why I have indicated that what they've used is a weak acid, is because you cannot use something so strong that you're going to kill the bacteria. You're going to dismember the proteins that make up the flagella, because आपको सब कुछ intact रख के सिर्फ और सिर्फ प्रोटॉन ग्रेडियंट्स को थोड़ा सा चेंज करके देखना है कि फ्लैजुलर रोटेशन कम होता है या नहीं, शायद बंद ना हो, बट एटलीस्ट अगर कम होता है तो उससे ही हमें ये पता चल जाएगा कि the translocation of the protons is directly affecting the movement, or it's directly causing the movement of the flagella. But if you think of adding protons that brings it down even further, 5.5 se niche, 4, 3, 2, 1, your cell will not be there. The experiment wouldn't work for sure. Now, if you bring it down to 5.5, low pH, and now is there a mechanism inside the cell that can recover the pH? So anything that has to recover the pH back to say normal of about say 7 or even 6.5, 6.8, that has to be an active mechanism of proton transport.
Yes, ma'am.
Right? Because then you're going to be translocating protons against the proton gradient. अभी सेल के अंदर ज़्यादा है, सेल के अंदर से बाहर आपको push करना है. That has to be an active mechanism of transporting of proton gradient, and that would have to be definitely dependent on ATP. In fact, what we are going to do in the next class is look at the ATP synthase system, and so we will see that this motor that drives ATP synthesis, if it is run in the reverse direction, it can hydrolyze ATP and push protons against the proton gradient, and those are also present on the bacterial membrane. Those are also present on the bacterial membrane. Whether bringing the pH inside the bacteria to 5.5 would cause these protons to be pumped out using these ATP synthase system, um, you can just guess. I mean, in this experiment, there's no way to understand that. You have to design some other experiments to understand if those ATP synthase systems are becoming active and they're pushing protons out. Possibly yes. Possibly yes.
Okay, ma'am.
But everything here depends. Your question was good. Your question was good, but you see the reason why I've mentioned sodium benzoate is exactly this. You do not want to bring the pH so low that you're disturbing the entire cellular system. Again, the reason why the pH was brought back up is again to show that you've not disturbed it to the point of irrecoverable damage. That you've just blocked the translocation of protons and you've not made the flagellar system non-functional by damaging the subunits or dissociating subunits or something like that.
Okay, ma'am. Thank you.
Thank you.
Ma'am, I have the same question. Like, in nature, we have bacteria, and outside the cell, there is higher hydrogen ion concentration, and inside the cell, the hydrogen ion concentration is lower. That's why the hydrogen ion goes from outside to inside. But in nature, there will be a point when the hydrogen ion which is going from outside to inside, both will be at equilibrium. If it keep going, then it will be like inside hydrogen and concentration will be the same after some time. So after that, flagellas will not rotate. So like bacteria will cannot move. So there must be like a mechanism uh, which keeps hydrogen and pumping out of the cell. I guess this was the same question he was asking.
Yes, but when you're talking about bacteria, we say that we harbor millions and billions of bacteria. And millions of billions of bacteria in an environment are seeing, I don't know, trillions or even more amount of these ions. I mean, how many amount of ions they're seeing? They're seeing the entire environment, right? So when you're thinking like this, don't think of it in a limiting quantity. Even जब आप LB में grow करेंगे, you add so much of sodium ions. You add such a lot of sodium ions that it is enough for generations to grow and for generations of bacteria to propel their flagella. So let us not think that those ion concentrations in the environment would be limiting. The ion concentrations are not limiting. Compared to the number of bacteria, the number of flagella they have to propel, the ion numbers are actually tremendously huge. Huge. So I would suggest not to think of it in this way. But of course, there are mechanisms to drive protons or drive ions out of the cell. Against a concentration gradient would always require energy. But don't think of it this way, that there is limiting number. This, it is not. Outside the cell, it definitely has uh, huge numbers of these ions for translocation at any given point of time, no matter how much the bacteria multiplies.
Yes, ma'am, understood, understood.
Ma'am,
Yes, Sarika.
ma'am, is this always a 40 plus 15-degree movement?
No, no,
Like
no, 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 right. You can also have a backward movement. I did not show that slide here. It's part of the same experiment where they showed that the flagella could take a backward step. No, not a backward step. That's a counterclockwise motion. I'm not saying that. What I'm saying is plus 14 डिग्री जो move करता है वो कभी कभार minus 14 डिग्री भी दिखाता है. ऐसा नहीं है कि नहीं दिखाता. But that is like a really, really small part of it. Let me share the screen once more so that it's visible. See that all of these steps are actually going up, but if you see here, maybe there's one step that goes back. So it's not that it does not take maybe one or two steps back, those 114-degree angles सामने जाने की जगह पीछे. That's a rare event. That's a rare event.
okay, ma'am. Majorly, it's building it up.
Yes, yes. But the movement of the entire motor can be clockwise or anti-clockwise. That could happen, and that happens. It depends on the clockwise and the anti-clockwise motion, whether the bacteria is propelled in one direction, there's bundling, there is a run motion, or there is a tumble motion. So that happens. So the entire motor can go clockwise or anti-clockwise. But while it is going clockwise, is there a point where it just takes 114-degree back? It is possible, but it's a rare event. It is possible.
Okay, ma'am. Ma'am, I have a doubt in many project.
Yes.
Second question, we have to do multiple sequence alignment for kinase gene. So in humans, we have 48, 45 kinases. So we have to do it for all of them. And they also have the transcript variants.
Oh, okay, okay, okay. So for the transcript variants, I would suggest do not take all the transcript variants. You can take any one of the transcript variant. Because if a gene has multiple isoforms, multiple transcript variants, just take any one of them. Do not take all of them. If your program doesn't allow you to do sequence alignment of say more than 10 sequences, then you limit it to 10. 10 करके ही दिखाइए.
Uh, 10 price of
If it limits you to not more than 10 sequences or not more than 5 sequences, then you limit it to 5 and show those 5 in blocks of 5. For those genes where you have multiple transcript variants, multiple isoforms, just take any one. Any one. The idea is for you to understand, okay, what we have read is actually what you're getting when you do these alignments.
okay, ma'am. And we have to clear N kinesins, C kinesins, and M kinesins, and then do the MSC or all of them together. Like for M kinesins, one multiple sequence alignment, and for C kinesins,
You could do
different
it
for
that.
M
You could do it that
kinesins.
way. If you find that even while doing the N kinesins, you feel that there are too many and the sequence alignment is not allowing you, you can break it up into 2 groups, 3 groups, and do it. The idea is this, that you should be able to understand that okay, this is one domain component that is coming repeatedly in all these kinesins. Is there such a domain component? What is that domain component? This is the idea.
Okay, ma'am.
If
Thank
you
you.
can answer that question, whichever way you're doing the sequence alignment, that's fine.
Okay, ma'am.
Any other question for the mini project? If any of you have any difficulty, I'm sure many of you might be around on campus. If you have any difficulty, just you can come over and talk. Same goes with Farman. If you're around, you can come over and talk. We are always available in the labs. Um, so, all right.
Okay, thank you.
Thank you. Ratnesh, I think you're also on campus. If any of you need any help, you can always drop in and we can discuss. All right, students, if you have any more questions, you're free to write or you're free to even drop in. As I said, we are always available in the lab or in the office. And see you on Friday. We also have a class this Saturday. So see you. Bye bye.
Thank you, ma'am.
Thank you.